Hello and welcome to GCSE exam questions, and the topic is infection and response. So all these questions are taken from real exams. Okay, so let's start from some examples from exams. So chlamydia is caused by a pathogen. So pathogens are could be bacteria, viruses, protists, and fungi. Okay, so chlamydia is transmitted by insects, sneezing, sexual intercourse, or contaminated food. So in this case, chlamydia is a sexually transmitted disease. So I'm going to put my pen on here. Let's have so it's sexual intercourse. The type of pathogen called is chlamydia. Yeah, so we just actually just talked about this, didn't we? In this case, chlamydia is actually a bacterium. So that's a singular of bacteria, bacterium. Okay, so sort of a bit of a bonus content. Let's think about other illnesses that are caused by other. So bacterium, you could have things like salmonella. Okay, you could have, like I said, like chlamydia. So fungus, usually in humans, these are not serious infections, but they can be. Athlete's foot. You get something called ringworm. The only protist we do at GCSE is malaria. Virus, we know COVID. Other COVIDs. You have virus of measles. Quite a lot of viruses, colds and flu. Okay, there's other ones here that we'll talk about those. So yeah, so we'll get on to other questions, but there's some examples. So figure 3 shows the number of cases of chlamydia in the United Kingdom per 100,000 people between 1996 and 2013. So you probably think, why is there maths in a biology paper? It's because there's always 10% maths. You're always going to get some calculations, usually to your population, things like this is, populations of other animals, bacteria, could be humans. There has to be some numbers in it. Okay, so you've got cases of chlamydia per 100,000 people and the year. Describe the trend in the number of cases of chlamydia between 1996, so it starts, and 2013, so where it ends. So actually, it reaches a peak between 2010 and 2012 and then goes down. So there's 2 marks here, so we need to describe 2 things. So cases of chlamydia. So these are obviously ones only known because these people visited the doctor. Probably more than this. Case of chlamydia rose between 1996 and let's go for 2000. Looks like 2011, doesn't it? And 2011. Okay. After 2011, cases started to fall. Okay, so state the number of cases of chlamydia per 100,000 in 2013. So we just need to read it from the graph. There's only 1 mark. So let's go across. Okay, so it could be quite hard to read. It's there. Okay, so let's see what this is going up in. 1,500, 6, 7, 8. Yeah, it's only going up in 100s. This is quite easy enough. 1,500, 1,600, 700, 1,800. We just had to read it from the graph. Okay. The population of the United Kingdom in 2019 was 64 million. Calculate the number of people with chlamydia in 2013. Okay, so it's a maths question. Right, so we've got 1,000, so per 100,000 people, 1,800 had chlamydia. Right, so there's a few different ways we could do this. Okay, that's 64 million. So what we could do, we could times these both, okay, to get times this to get to 1 million, then we times it by 64. I think that's the easiest way of doing it. So 100,000. So all we need to do is times it by 10, okay, to get a million. Okay, so you still have 6 zeros after it. Okay, so timesing that by 10, that gives 1 million. 2, 3. It's probably standard form, isn't it? All zeros. That times 2, oh yeah, equals, sorry, is 18000. Right, now we need to times it by 64. Right, so we're just going to get that number there. Right, so this is a bit we can do on your calculator. Okay, you're allowed a calculator in this exam. So 18. Delete. I was just calculating something else in the exam. So 1800. So 18,000 times by 64. Have a look. So 1,800. 8 times. Okay, so we've got, it's quite a big number. 1,152,000. Okay, 152. So that's quite a lot of people, isn't it? Okay. So yeah, because I remember even I think when I was at school, not mentioned as much now because we see that cases fell, but I think people receive more education about how it's passed on. So I was people talking about, yeah, it was quite a big problem back then. And people, I think, yeah, the education they received, because sometimes you might say something like, why did it fall? You say, well, there could be people better educated about how it's passed on and awareness of it. So that could be any sort of transmitted illness like that, isn't it? Sort of thing, because it's sexually transmitted. So it's not like a cough or a sneeze one, but we know from any sort of transmission diseases that it can be sort of by education, we can actually cut it down. People get a bit more formed. Okay, so chemicals can be extracted from plants. Some of these chemicals kill bacteria. Ooh, antibiotics. So that could be used to treat chlamydia there, because that's why it's linked to the question. A scientist spread some bacteria on a nutrient agar plate as shown in figure 5. What is being shown in figure 5? Okay, so it's not selective breeding, because that would be an animal or a plant. Genetic engineering? No, you need a bit more precise things than that. Cloning? It doesn't show anything with cloning, does it? So it's aseptic technique. Explain why the scientist worked near a Bunsen burner. Right, that's to so it's to disinfect. Okay, so to use the flame to disinfect the loop. That is called a loop. Okay. Well, if you forget that, just put effects equipment or so piece of equipment to spread the bacteria. If you describe it, if you got a word, don't panic, just describe it. If you describe well enough, you'll give you marks for it. Okay, a scientist spread bacteria onto the surface of 2 agar plates. A filter paper disc was placed in the center of each plate. Each filter paper disc had been soaked in a different chemical extracted from plants. The results are shown in figure 6. There's chemical A, zone of inhibition. That's where bacteria were killed or didn't grow. So there's no bacteria here. Okay, they died or they just didn't grow. Zone of inhibition 8. So that's got a bigger zone of inhibition. That's got a bigger area where bacteria didn't grow. The area of a circle is calculated using pi r squared. Calculate the area of the zone of inhibition for chemical A. So we're just substituting numbers into a formula. So pi, it says use 3.14 for pi. So you've always used in maths all the time. 3.14, but remember to square it. So 12 squared. So we're going to get 3.14 times 144. And think about the units, or millimeters squared we're going to get this. So let's get a calculator out again. So we've got 3.14. So if it tells you to use pi as that, do use that instead of the pi button if it's telling you to do that. Okay, so we've got 452. Oh, I've lost my place there. 452.16 millimeters squared. Okay, for that one. So that's just a calculation there. Let's get on to the next thing. So the scientists concluded that chemical A was more effective than chemical B at killing bacteria. Give two variables the scientists need to control to make sure this conclusion was valid. Okay, so you need to have the agar plates had to be in the same conditions. Okay, so if one was in a really hot place, one was in a really cold place, so same temperature, light, etc. Okay, and also, was the same type of bacteria. So it might be good at killing a certain type, not another type. So same type of bacteria was on the agar. Okay. Clostridium tetani is a bacterium that can be found in soil. It causes the infection tetanus. You've probably heard of that. Children are vaccinated against tetanus. Explain why these children do not get tetanus if the bacteria enters the body through a cut in the skin. So it's how vaccination works. So a vaccination, that's how all vaccinations work. So a vaccination introduces a dead or weakened bacterium. So the one that's C tetar, yeah. Okay. It's E, isn't it? Yeah. C tetani. So that's a short version of writing that. Okay. Into the body. The immune system produces antibodies to fight the specific pathogen. Okay. If the body is invaded by C. tetani through a cut, the body has antibodies ready to fight and kill the bacteria. Okay, same with the COVID vaccines. That your body, if you have a vaccination, your body makes the antibodies for that strain of COVID. I know some people talk about different strains of it. Okay, for that strain of COVID, and then if you actually your body does get invaded by the virus, okay, then it's already knows how to fight it. So it's already made specific antibodies to fight that virus. And okay, so vaccinations are very effective. And some illnesses, like I've had smallpox, has been actually eradicated by it. But some people, obviously, who are saying that they don't believe vaccines work, that some illnesses are coming back in some parts of the world, obviously, because they're saying they don't work, and then illnesses only because people weren't scared of certain illnesses because obviously it was much lowered by vaccinations. Okay, so actually they do work very effectively. Okay, colistin is an antibiotic used to treat infections in the bloodstream. Some bacteria are resistant to colistin. Explain how these bacteria have become resistant to colistin. Right, okay, so it's talking about antibacterial resistance really here. So colistin killed most bacteria, but some bacteria will have a mutation, have a natural mutation that makes them immune to colistin. So these immune bacteria survive and go on to reproduce to produce more bacteria with the immunity to colistin. So you could say obviously overuse of antibiotics will create more resistant bacteria. Because what you're doing there, you're eliminating ones that are not immune to it. And if you leave the ones that are immune to it, they'll multiply and create lots of ones that are immune to it. That's why doctors don't want to overprescribe antibiotics. You get bacteria that are immune to antibiotics. You just get stronger and stronger bacteria that are immune to it. That's why you don't want to prescribe it too much. Okay, carbon dioxide is carried in blood plasma. Human blood also contains red blood cells and white blood cells. Explain how the structure of red blood cells and white blood cells is related to their function. Okay, so we've got two things there. Okay, red blood cells. Okay, so be quite artistic in the door. Red blood cells. So they are shaped. Like this is like looking down, and then side on, they've got like a squashed middle. Like a bone as well. A squashed middle. Okay, so structure. So they have no nucleus. So again, it's good to draw diagrams. Drawings of diagrams, and they're sort of like call it a dimple, sort of like a sort of squash bit, like a sort of it's called a biconcave shape, squashed in the middle. So no nucleus. So no nucleus and biconcave shape to carry more oxygen. So red blood cells, also red blood cells are red because they contain oxyhemoglobin. Oh, no, just hemoglobin, sorry, and it binds to make oxyhemoglobin, so hemoglobin. Which binds with oxygen to make oxyhemoglobin. So we've got white blood cells. Okay, so we've got two different types of white blood cells. We've got the phagocytes. And we have the lymphocytes. Is that COVID? So phagocytes, so ingest, these are part of the immune system. Both white blood cells. So phagocytes ingest pathogens, so killing them. So lymphocytes, so produce antibodies specific to pathogens. So these often look like little T's, often look like little tags. You've got a pathogen. Try a different colour. Got a pathogen like that. Then the antibodies will actually attach themselves. Okay, they'll sort of like tag them, because one thing they'll do is they'll tag them so that the phagocytes will actually notice that they are an invader organism and kill them. So it's pathogens to kill them. And they also produce antitoxins. Okay, so because some bacteria produce toxins. Okay, so thank you for listening to the video and good luck in your exams, and hope to see you in a future video. Thank you, goodbye.